Please welcome Buzz Palmer, MedTech Actuator.
Ladies and gentlemen, hello from Singapore. My name is Buzz. I am the CEO of MedTech Actuator. It's my great pleasure tonight to introduce the brilliant Stefan from DermR. We nominated the startup because they are transforming how we diagnose skin cancer. Instead of painful and costly biopsies, where more than 60% turn out to be benign and avoidable, DermR has developed a painless microneedle patch combined with a qPCR test. The team already has incredible, impressive traction. Why should they be the falling wall science breakthrough of the year? Because they are breaking the wall of invasive skin cancer diagnostic. Their biopsy-free point-of-care solution empowers GPs, nurses, and even aged care providers to detect and subtype skin cancers quickly and equitably. A true diagnostic platform with the power to transform skin cancer detection, diagnostic, and treatment worldwide. Please join me in welcoming the outstanding and genuinely nice guy, Stefan.
Breaking the wall of biopsy-dependent skin cancer diagnosis. Stefan Maisie, DermR Health.
Many of us here have faced it. That uneasy feeling when you first notice that suspicious spot on your skin. What do you remember about that feeling? Did the thought that you may need a biopsy delay you from getting it checked? Did you require one anyway? Was it painful? Were you left with a scar? Skin cancer stands as the leading global cancer, a hidden burden costing every healthcare system billions of dollars to manage. Yet, despite decades of medical innovation, diagnosing skin cancer still depends on an invasive surgical biopsy. Our research has found that over 60% of skin cancer biopsies are benign. In Germany, there was more than 1.2 million avoidable procedures, costing the healthcare system over 240 million euros just last year. And as for melanoma, it takes on average 15 biopsies to find a single melanoma. Now, while my home country Australia may lead the world per capita, the remaining countries are all cold climate nations, challenging the assumption that melanoma is associated with warm, sunny climates. Today, if a patient suspects skin cancer, they have to see a doctor for investigation. If cancer can't be ruled out, they either undergo surgical intervention or referral to a dermatologist. However, due to workforce shortages, to get a doctor's appointment can take weeks, securing a dermatologist often months, delaying diagnosis. Now, while biopsy and histopathology remain the gold standard for diagnosis, it's that clinical decision pathway, that decision to cut, that creates that bottleneck where over 60% of lesions excised are benign. We're on a journey to replace 1 billion avoidable skin biopsies. Now, I know firsthand that delaying a diagnosis is significant. In fact, 2 of those photos are of my mum, who's the reason why I started this. By leveraging advancements in digital genomics and advanced manufacturing, we're developing a new way to diagnose skin cancer. The DermR patch is a first-of-its-kind microneedle patch comprised of uniquely barbed structures that can painlessly extract viable skin cells without the need for surgical intervention and at a lower cost. The sample can be sent to any pathology provider for a standard qPCR analysis. Now, while other tools can only highlight suspicion, our proprietary genetic test can provide molecular confirmation and uniquely subtype skin cancer, distinguishing between melanoma, BCC, and SCC using just a few cells, enabling earlier, more informed clinical decision-making, including whether to proceed with surgery, monitoring, or non-surgical treatments like new topical immunotherapies. Now, in most countries, only doctors can perform biopsies. Our solution enables nurses and allied health to administer a biopsy-equivalent diagnostic test for the first time, unlocking a globally underutilized workforce and expands frontline cancer detection to underserved regions. Dermoscopy, light-based tools, AI, they all aim to improve triage, but they all have the same limitations. They can't subtype skin cancer, they fall short on specificity, which is the ability to rule out non-cancerous lesions, and they still rely on an excision for confirmation, compounding the existing burdens. In our first-in-human clinical trials, DermR detected skin cancer with 99% sensitivity and 96% specificity for ruling out non-cancerous lesions. By intervening before the decision to cut, DermR outperforms the current clinical workflow and all triage technologies. Our revenue will come from selling our next-gen patches and qPCR kits to pathology providers, enabling integration into their existing clinical networks. Everybody benefits. More patients can be seen in less time. Genomics increases efficiency, reduces human error, which increases margins. In the last year and a half, we've completed our first in-human clinical trials, received national attention for our innovation, and next month, we officially start our pilot phase trials. And we're raising 3 million euros to help us finish our pilot, getting us closer toward regulatory approval. My name is Stefan Mazy. I'm the CEO and founder of DermR. 15 years ago, I pioneered the consumer skin genomics industry. Today, I lead a team that are all experts in their own right, but it's their individual stories with cancer that make this more than a job, but a mission. Now, we often hear about the need for more doctors, or the need to invest billions in new infrastructure just to reduce wait times. But what if we removed millions of patients from the system that don't need to be there? Can you imagine what all healthcare systems would look like if we broke the wall to biopsy-dependent skin cancer diagnosis? How many lives do you think that would save? This is how I believe something the size of a freckle will change healthcare systems and impact the lives of 1 billion people, one pain-free patch at a time. Thank you.
I cannot applaud, but I would applaud, definitely. Thank you so much. Wow, okay. First question, second question. Thank you.
What makes you so unique that you are the only one doing this successfully?
Yeah, so it really comes down to a few things. Number one is we've figured out a scalable way to manufacture barbed microneedle structures. So there is research out there about extracting tissue. They're all prototypes. There is no scalable method, and we've figured out a solution that can do it. Number two is we've applied that to a genetic test, which we have IP around as well. So it's not just one thing, it's the manufacturability of the microneedles at scale for cheap. It is our genetic test component. It's also the trade secrets around how we do it that make us successful.
Detlef, please.
What's your commercial route? Do I go into CVS and buy the kit and do it myself and send it in and pay it out of the pocket? Or do I need to see a physician or a dermatologist?
So our current reimbursement strategy, this is a global problem affecting every single country. We've made the decision that this is not an out-of-pocket. This is something governments need to support. Our reimbursement structure, we've identified over 30 innovative payment streams across Europe that will allow national coverage using certain Medicare item numbers. We've also identified something similar in America, making the entire system completely reimbursable under major healthcare systems.
GP signs off.
A GP will sign off on it, yes.
Andrea, please, you're the next.
Yeah, thank you. There are already some punch biopsy devices on the market. What makes you different to those that are already existent?
Yeah. Look, I guess the main thing that makes us different is functionality and workflow. So you're right, there are punch biopsies out there. The problem is when you actually see what it takes to extract the sample in the lab workflow, it is very, very fiddly. A lab can't physically do 1,000 of them in a day. The way that we've developed our workflow as a PCR test is fully automatable. So from a pathology perspective, their bottleneck is histopathology is quite laborious. All of this can be automated, reducing the need for lab technicians. And that's probably one of the biggest strengths that we have, that it's a plug and play for pathology without them needing to integrate or hire new staff or do something new.
Next question. Even the audience may ask. Andrea again, please go on.
Yeah, I have some other questions. For melanoma, there is the need to know the thickness. So you only say yes or no, it's benign or malign, but nevertheless, there are different areas and clonal expansion areas. Do you use this biopsies kind of multiple on one lesion, for example? Because also for basal cell carcinomas, you know, there are different areas, and it's very important to get the right one.
Correct. So what makes our patch quite unique is the depth. So the microneedle patch itself is 1 millimeter long, and along the patch there's unique barb structures. So when it goes into the skin, it actually collects tissue from all the layers of skin, so we can ensure that we're collecting the viable sample for that type of lesion. So it's not designed just for melanoma, it's designed to figure out what type of cancer it is, period. So that's the yes-no pathway. And then the uniqueness is our ability to subtype using our genetic tests. So one, if it is cancer, step 2 is what type of skin cancer. And so we know that certain types of BCC, you don't need that excision, you have topical immunotherapies. If it's melanoma, you don't need the biopsy, you go straight to urgent care. So that's where we're sort of able to triage and effectively manage skin cancer.
Thank you. Do we have a question in the last row there from the youngest member of the audience? No? We're good? Okay, because we have only 10 seconds left. No, I'm sorry. We have 5 seconds. I'm very sorry.
All right.
Super interesting, but you may ask, definitely. May I have a look at it very quickly?
There.
Oh, nice. Thank you very much.
Thank you.